Hi, I'm Bin Ren. I'm an associate professor of surgery and biomedical engineering at the University of Alabama at Birmingham School of Medicine. Our laboratory is interested in endothelial cell biology, functional angiogenesis, and arteriogenesis in the context of ischemic cardiovascular diseases and cancers. One of our research focuses is on epigenetic and transcriptional regulation of endothelial cell transdifferentiation and angiogenesis. To understand the transcriptional mechanisms of angiogenesis, we have established transgenic engineered mouse lines in which we have not only deleted the specific genes associated with angiogenesis in the vascular endothelial cells, but an enhanced EGFP probe will be genetically tagged onto a ribosome subunit of endothelial cells. In this way, we can isolate and purify ribosome-associated messenger RNA from endothelial cells in different tissue organs directly in transgenic animals. We can then use the purified messenger RNA for downstream analysis of transcripts and the transcriptome by RNA sequencing and real-time qPCR. In this video, my students Patrick, Moran, and Jordan Palmer, and the research technicians Nicholas Barnekow and Rong Yuan will show you an approach to genotyping transgenic mice and isolating messenger RNA directly from the endothelium in the transgenic animals.
Genotyping is an important preliminary step in this protocol to ensure that the mice have the GFP tag on the ribosomes of endothelial cells. Since all downstream RNA isolation steps rely on the presence of GFP tagged ribosomes, it is essential to determine the genotype prior to beginning the